Hello everyone, a very good evening. After having spent so much time with good amount of time with peptide sequences, we've named our group peptide sequence BAHS, which also doubles up as an acronym for our names. We would like to thank Professor Sergi and Life Story Health Inc. for giving us this opportunity to analyze this data set. Now diving into the presentation. Now, the objective of this analysis was to identify trends, both internal and interactive, within the variables for different patient profiles that were given to us and the other variables within the data set. We also wanted to analyze and understand how these variables interact with one another and the role that they play in predicting modifications or post-translation modifications, which in turn offer a lot of insights into disease progression and prevention. Now, going into descriptive analysis, what we've done is we've drawn up a descriptive analysis for the all details data set that was given to us. Now, this is a univariate analysis of each and every variable that was present in the data set. As we can see here in the observed mass section, we have a range of 0 to 4,000 in terms of observations. And in observed retention time, again, it follows, as we can see, more or less a normal distribution. Now, this univariate analysis helps us just understand the nature of these variables and then understand the domain better, and which is why we started off with this analysis. Uh, Again, charge, if we see the charge histogram is kind of skewed to the right and mostly has observed values of 0 to 4. The observed mass and parent ion mass follows a similar distribution. Parent ion mass is something that we've calculated separately. It was not given to us, and we'll get into that during the course of the presentation. The modified mass, again, which is also something that we arrived at later, is which is the difference between observed mass and parent ion mass. Uh, Again, very closely resembles the distribution in the parent ion mass, which could point to the fact that there have been substantial modifications observed in the samples that we were given. We also drew up box plots of these different variables to detect outliers. Now, again, if we see the observed mass, it has a set of values uh, concentrated in the range of 9,000 to 10,000, with a few outliers observed about 13,000. In charge, we can see a median value of 2, with most of the values concentrated between 1 to 4 and an outlier at 11, because charge only had a fixed number of values in it. And in the assigned intensity, we see a lot of zero values occurring, with the maximum value again cropping up to 100. So the dispersion of the data was pretty high. And this could point to some amount of, like, a great deal of fragmentation of ions and their differing peak intensities. Moving on. So here, like I was mentioning, we have added these two variables in the all details data set. One is modified mass and one is parent ion mass. Now, the reason that we felt having the modified mass could help us unravel some more insights was because when you start comparing these modified masses with standard modifications, they actually give you a window to understand PTMs, and therefore we calculated modifications for these peptide sequences. And some of the steps that we took for this calculation was, one, to install a package called the ProtWiz. The ProtWiz is actually used to perform quality checks and visualizations in analyzing mass spec data. And this package was tested and developed at the Functional Genomics Center in Zurich for the first time. We calculated the parent ion mass here using the parent ion mass function that is a part of this package. Next, after we calculated the parent ion mass, we were able to then calculate the modified mass, which is the difference between parent ion mass and observed mass in Daltons. And like I said, when we are, and like we were also told in the proposal, once these modifications, these calculated modifications, once we start comparing them to standard modifications, we can identify PTMs for each of the patient IDs, and that could give us a lot of insights into their health. Next, we wanted to understand interactions and correlations between each of these variables. And so we adopted this using this correlation matrix. And as you can see here, uh, any any point here, any number here that's close to 1 expresses is an indication of a strong correlation. And we can see here that the observed mass and modified mass again has a decent significant correlation at 0.65. uh, Charge and observed mass also has a good amount of correlation at 0.93. Parentine mass shares a significant correlation with modified mass at 0.60. Next, we wanted to build a model using these variables. Uh, and since modification mass is one of the most important variable in predicting the nature of a peptide and PTMs itself, we tried to build a model with the variables that affected most strongly, and we identified these variables from the correlation matrix that I just spoke about. And this is the linear regression model that we've built here. Uh, and we used observed mass and charge as our dependent variable, and modified mass here was our independent variable, and we arrived at an adjusted R square of 0.43. The strength of the model is not very high, which indicates that these two variables do not play a significant role in affecting the modified mass, and maybe uh, while these variables individually may be affecting modified mass and they may have a strong relation, but when they are working together, they don't really influence the modified mass that much, and that is something that we realized after running this model. Moving on to the consumer health data that was given to us. Uh, Here's some descriptive analysis that we've done, and what we found was that brand Tampax and followed by Kotex and Playtex are the most frequently used products among within these patient IDs. Next, if we look at their menstrual health, most of them have responded that they have a moderate flow. Now, a lot could be done with this data set because the proposal also said that this data set and the analysis that we draw from this data set could affect corporate partnerships. But what we felt was that we were missing some data here. Now, we were missing data on observed mass and modification for these patient IDs. And having access to that could help us explore the relationship between the menstrual flow and the degree of modification, and then eventually the type of products that they used, which was an important variable in this data set. And again, we had a lot of missing values within this data sets. In terms of bath soaps and detergents and wipes, these were again some variables, and we didn't have access to which bath soap that particular patient ID was using or which detergent, and it had a lot of missing values. Now, having that data could have enabled us to identify trends at a category level and see how different products perform or interact with the health parameters again that were given in the data set in terms of menstrual flow, cramping, and abnormalities. Also, understanding the frequency of usage of these products could also help make comparisons between heavy, light, and moderate users of this product. Like for example, if we have to draw up a hypothesis that patient IDs that used Tampax most frequently were more likely to have a heavy menstrual flow or otherwise, or were most likely or in their patient IDs, more PTMs were observed. So if we had that data as well, we could kind of make more definitive conclusions on the relationships between the products they used and their health parameters. Next, another task that we were given was to identify proteins from different peptide sequences. So there was a sheet that was given to us that had unlabeled data. And then there was a sheet where we had protein names and known protein names and their masses. Now we first calculated the masses of this unlabeled data using the MZ formula. And what we learned that the masses of these in these two data sets were drastically different from one another, like in terms of range. So if the mean value of one data set was 460, the mean of the other column, the mass column of the other known sample was 92235. So none of these protein masses were matching with each other, and we also felt that since proteins are made of peptides and a lot of complex amino acids, and there are a lot of complex interactions between them, this way of just comparing the masses was kind of too straightforward and not very sophisticated to arrive at protein names. So, which is why we tried to identify a function within the package that we're using, and we bumped into this function called the fragment function. Now, what we did was that since we had the modified mass of these, we had calculated the modified mass of these protein samples, we identified 3 peptide sequences from within the data set that we were given, and this is the all details data set that was given to us. And we took the minimum modified mass, the maximum modified mass, and the median modified mass. Next, we used the fragment function to pick these peptide sequences with the minimum, maximum, and the median modified mass. And this is the result that it produced. Now, basically, these are the 3 peptide sequences, and the fragment ion mass function has essentially broken down these peptide sequences into different ions that make up this sequence and their masses. And there are some ions that appear more frequently than the others, and they've all been color coded. And the ones that appear most predominantly are given by blue and red, and they are termed as B and Y if we go down to the scientific literature of it. Now, when we backtrack these masses, the masses of these ions, we are able to identify the different combination of amino acids that make up this peptide sequence, and this in turn will help us identify the protein and their modifications, and that's how we can backtrack this peptide sequence to the protein that it must have originated from. And in scientific terminology, this process is known as peptide mass fingerprinting. Uh, and this is an example of how we could arrive at protein masses and identifying the respective proteins. And that's all that we have for you today. Thank you so much again, and we are looking forward to hear your comments.